All right. Hi, everyone. Welcome to Science and Nature Untapped. So you are at the right place. We are going to wait a few seconds to allow everyone else to join us. So the theme of the talk today is the FINS project, Little Fish, Big River, who will be presented by Matt Wendell from the St. Lawrence River Institute. My name is Yannick Rozo. I work for the St. Lawrence River Institute, and I am replacing Stephanie Hildebrand today as the host for Science and Nature. So we'll get started. So a few housekeeping items as before we continue. So if you have any questions, we would invite you to please put them in the Facebook comment section, and we will get to them during the question and answer period. You are also welcome to share with us where you are coming from today. Seeing as this is a virtual event, we tend to have people from all over, and it's quite nice to see where people are coming from. So this is Science and Nature Untapped, which is a virtual free monthly speaker series from the St. Lawrence River Institute. It is being shared both on Facebook Live as well as through your TV, Kojiko. And so I'm just going to give a quick countdown to Gabe from your TV so that they can get started and also broadcast it through their channel. So Gabe, here it is, 3, 2, 1. All right, thanks everyone. Welcome to Science and Nature Untapped. So before we begin, we would like to acknowledge that many of us are living on the unceded land and traditional territory of Indigenous peoples. We're very grateful for the opportunity to live where we do, and we do thank all the generations of people who have continued their responsibilities to Mother Earth since time immemorial. I myself am coming to you today from the traditional lands of the Haudenosaunee, and given that this is a virtual event and we are being, we have participants joining us from all over, if you would like to find out which traditional lands or territories you are residing on, then we would invite you to visit www.native-land.ca. The River Institute has a few different social media sites if you would like to check them out or connect with us. Our Twitter account is called River Institute C, as is our Instagram account, so River Institute C. Our Facebook account is called the St. Lawrence River Institute, St. Lawrence River Institute only. And we also have a Future Climate Leaders Facebook account, which is FCL613. In January, we have another Untapped speaker, which will be Dr. Courtney Holden from the River Institute. So this is happening on January 5th, and the theme of her talk is The Secret Life of the American Eel. So we would love it if our viewers could join us for that event as well. Another upcoming event that we have is another Great River Rapport workshop, which will be led, it's a presentation by Abraham Francis, and it will be on connecting with community participatory research approaches. So this is happening next Wednesday, December 15th from 7 to 8 p.m. And it is also going to be a Facebook Live event. If you would like to donate to the River Institute, you can do so at riverinstitute.ca/donate. And this would be making a donation to the River Institute Foundation. All right, so now to get started. So a little background on Matt Wendell before I pass it over to him. So Matt Wendell is a biologist at the River Institute with experience in environmental science, freshwater ecosystem ecology, spatial analyses, contaminant analyses, and remotely piloted aircraft systems, RPAS. He completed a Bachelor of Science in Environmental Science from Queen's University and a Master's of Science and Biology at Memorial University in Newfoundland. In addition to his work as a research scientist at the River Institute, he is also a professor in the Environmental Technician program at St. Lawrence College. Matt has a certifications for open water scuba, the Ontario Stream Assessment Protocol, OSAP, and Royal Ontario Museum. So without further ado, here is Matt Wendell.
Thanks, Yannick. I'm just trying to share my screen. A little difficulty here. Oh, there we go. Wonderful. Thank you for that intro. And hello to everyone that's tuning in right now. I'm Matt Wendell. I'm a research scientist at the River Institute in Cornwall, Ontario, and I'm really happy and excited to be here today to talk to you about a project that's near and dear to my heart that I've been working on for 7 years now, almost the entire time I've been at the River Institute. And it's a project that's grown in scope and partnerships and results, and just I'll be highlighting some of the work we've been doing related to this project. And the project is called the FINS project, and FINS is an acronym for Fish Identification Nearshore Survey. And the subtitle, I wanted to convey that it's a huge river. The St. Lawrence River is one of the largest rivers in the world. And we often think about the fish in the river as in terms of the sport fish, the larger fish that people like to angle and catch. And small fish and small fish populations are quite considered, even though in terms of abundance, they far outnumber larger fish, and they're the base of the food web. So it's really, really critical that we pay attention to that so we can monitor the health of the river that we love so much. So I'll just start here by, there we go. I want you to imagine that, I know it's December, but imagine it's back in July and you're on a boat, for example, you're just floating in the river, the St. Lawrence River, the water, you can clear, maybe you have a cold drink in your hand, it's just a nice breeze in the trees along the shore. And let's just linger there for a moment. You're in shallow waters and your gaze falls to the water and you notice lots of fish swimming in the shallows around you. And this happens for a lot of people. They just kind of casually notice these types of things. And maybe you wonder what they are. It's really hard to see them. They swim out of sight really quickly. They're very small, so it's kind of hard to see details about them. And if you wanted to know more about those fish, what would you do? Well, you need to get a little bit closer to them, wouldn't you? You need to bring them maybe into your world to have a better look. Or you could go snorkeling or scuba diving if you have that ability. But how would you capture them if you wanted to have a look at them? It's something to think about. Fish are fast. They're pretty wily. And if you did capture any, how would you have a look at them and be able to tell what species they were? You need some training to do that, right? You need to figure out, are they native species? Maybe. You'd maybe wonder what type of habitat they live in. You might consider and ask the question, there seems to be lots of them. Is that normal to be this many of these fish? Are they increasing? Are they decreasing? Are there more than there used to be? You start looking around the habitat and wonder about the plants you see. Are those native? Should those be here? Are they important to the fish? What else is living in the river, this bottom? And the fish you see in this video right now, these are young of the year largemouth bass that we quite often find in the shallow margins of the river. So there's an entire ecosystem beneath the surface, an entire world that's just out of sight and out of mind for most people. And this ecosystem is full of linkages and relationships that you find in forests and fields and other terrestrial habitats, but we just don't see it because it's below the surface. And this ecosystem is disappearing, and it is disappearing faster than any other type of ecosystem in Canada. And the causes are predictable. Development of shoreline habitats, runoff of nutrients and sediments, introduction of invasive species, overexploitation, pollution, the usual suspects. And we need to know more about these ecosystems before they change and before they disappear. Today, we're going to focus on the shallow margins of the St. Lawrence River. These are areas and allows plants to grow, which in turn provides a lot of habitat for species, most species of fish that live in the river at some point in their life cycle. Some types of these habitats, nearshore habitats, such as coastal wetlands that you see here, are so rich in plant species and other features that they are one of the most biologically diverse of any type of habitat you find in the river. And unfortunately, these types of nearshore habitats are the most affected by human activities, such as shoreline development, nutrient runoff. So in this presentation, I'm going to be talking to you about this project that we've been working on for the past 7 years. I'm going to bring you into this underwater world in the St. Lawrence River, and I'll talk to you about some of the research that we're doing to study these nearshore habitats. So this project began in 2015. And just to cue your memory, Justin Trudeau had just become Prime Minister. Hamilton debuted on Broadway, and I was sporting at the time a quite impressive beard at the time. I'm not sure why, but there I am. And this project began with the question, how are the fish populations doing in the river? And this was brought up out of community concern in Akwesasne and Cornwall. A lot of community members were saying that small fish populations seem to be declining. So at the time, the River Institute got together with the environment program staff at the Mohawk Council of Akwesasne, and we started to plan some research, and we decided to focus on these community concerns and start to do survey work of these nearshore fish populations in the area. And we started this project with a relatively simple approach. Our team was fairly small at the time, back in 2015. We had a seine net that you can see there, and water quality meters to measure water quality at the different habitats, some buckets, some bins, some nets. And we did our best efforts we could at the time with our small team in 2015. And at this early stage, I should mention that core funding from Ontario Power Generation and the Trottier Family Foundation was really critical. That helped launch this program when we didn't have any other funding sources at the time. And we had some really good results. We caught about 7,000 to 8,000 fish that summer. We surveyed about 50 sites in the area. And there's a lot of types of fish that were in this area. And this map shows all those locations kind of in the Cornwall Akwesasne section of the St. Lawrence River. You can see all the sites there. So that was a great success. And then building on that, in 2016, all the way to 2019, we were able to expand our survey based on the 2015 results. So we're able to show government agencies that we have the capacity to do this type of survey work, that there was a real need for this work to fill in knowledge gaps on not only just baseline data of what fish populations were there, but also what species at risk might be present, if species, how were the human activities in the area affecting these ecosystems? Could we learn from the results we were finding to develop ecosystem indicators to tell us about the health of the St. Lawrence River, which is really on the mind of so many people. So between this period of 2016 and 2017, we were actually able to acquire government funding, and this was primarily from the Ministry of Natural Resources and Forestry's species at risk funding. And this allowed us to focus on species at risk, in particular the Pugna Shiner, which is a species at risk. We were able to hire more staff. You can see in some of the images here, we've incorporated more staff from the MCA as well as the River Institute. And at this time too, we started to develop collaborations with other organizations that were also doing survey work on the river. So in this picture, you see Dr. John Farrell on the far right there. And Dr. Farrell runs the Thousand Islands Biological Station in the Thousand Islands on the US side of the river, and has been conducting nearshore surveys for decades, and has generated an amazing set and really developed a really good understanding of, in particular, pike and muskellunge population trends for the river. So we approached Dr. Farrell's group in 2016, and he was very kind enough to invite our team out to participate with some of his surveys, and we really got a good sense of the types of equipment that they were using, the methods, the seasonality of it, and how they were using those results. That really benefited providing guidance to our project. Back in 2016, we also approached Parks Canada. So Parks Canada conducts nearshore fish population surveys in the Thousand Islands and the Canadian waters, and has also been doing that for decades, really targeting sites where young of the year muskellunge might be found. So they had also developed over the years, a very good approach, and we worked with them to develop some of our own approaches based on the examples we're using, the mesh size, how we're documenting various habitat features as well. So 2016 was a great year for us to really develop some really key partnerships initially in the project, provided some great guidance. And I think Josh Van Weeren, you can see in this picture, and Dr. Farrell for their guidance at that time. So that was a good chunk of research, and we developed a lot of results that really provided much needed information to fill in knowledge gaps for the distribution of Pugna Shiner, which is a species of risk that I mentioned, but also other species of risk during this time. And the modern era of this project now that I'll be mainly talking about is 2020 to the present. And our techniques and our approach has really expanded during this period. So we're still conducting fish surveys, but we've standardized the approach a lot more than we have in previous years. And today, something like we go to the site and we measure a certain standard length along the shoreline, and then we do a series of seine hauls. You can see this net here in this picture along those lengths of the shoreline. And we do that approach at every site that we go to, so that we can compare the amount of effort we use to actually catch the fish. And you can imagine if you'd spent surveyed a lot longer length of the shoreline, and you caught more fish, that would be hard to compare to a site where you say you only did one haul, for example, guys, your effort. And we've been doing this in the previous era, but we really fine-tuned it during 2020 and 2021. And this fish seining allows us to fill in knowledge gaps on the abundance of different fish species in the river. So the species richness is just the number of species at a given site and their diversity. And diversity can be skewed towards one species or evenly spread between species. So that's an important thing to measure. We've also looked at morphometrics, so the size of all the fish we're catching, that we're sampling. And this survey work, I should mention, takes place primarily between June and August, mostly between July and August. And we've actually expanded that into the fall in recent years as well. Last year and this year, we were actually out fishing in October. At each one of our sites, we measure the aquatic variables in the aquatic habitat. So this includes the substrates. Are they sandy or muddy? Are they large boulders, rocks, water quality as well, but aquatic vegetation is measured. And in 2020, Dr. Marianne Perrons, you can see here, helped us develop a standardized approach to measuring aquatic vegetation at each one of our sites. You can see that little square floating in the water there. That's a quadrat, the standard size. So we've been using that to get percentages of different plant species within each habitat, and then we can kind of expand that and generalize to the entire habitat. Putting this since the beginning, but measuring water quality, this is such a critical component to fish habitats and ecosystem health. So things like temperature, fish are very sensitive to that. Some species can't survive in waters that are too warm. Dissolved oxygen is also really important. Fish have a lower threshold for that, beyond which they can't essentially breathe. Turbidity is water clarity, so that really does vary across the river in nearshore habitats. And water clarity is really important for sensitive species like the Pugna Shiner, that clear water, and other fish like northern pike, for example, that are visual predators. We've also been collecting water samples to measure nutrients, and nutrients are really key. We have a lot of runoff. There's a lot of agricultural development of land in the St. Lawrence River watershed, a lot of farmland. And when we have rain events or spring runoff, a lot of the nutrients that are put on those fields run into the St. Lawrence River through various tributaries. So there's these pulse events where after our melting of snow, you see lots of nutrients enter the river. The nutrients, of course, feed the aquatic vegetation and algal blooms potentially as well in the river. And certain types of algal blooms, for example, cyanobacteria or blue-green algae, can cause some very harmful scenarios for fish and humans as well. So nutrients really tell us about the productivity, how well the ecosystem can generate life, essentially. We also look at shoreline, and it's quite staggering how little shoreline is left that's in a natural state in the St. Lawrence River. There's large sections of it, particularly on the northern side between Cornwall, moving downstream to Quebec, that's almost entirely developed houses and businesses and cities. And that really does, it's an issue. And there's other organizations like Watersheds Canada doing great work to document the extent of that waterfront development. So we document that as well as part of our survey work. And you can see the plants along the edge of the river here. So we document, that's gone. So those plants right along the water's edge, we document what species they are, what types of vegetation. And in some cases, that type of vegetation is all invasive, or it could be in a natural state. And that really does also affect the aquatic ecosystem that's immediately adjacent to those shorelines. We started also using, and this is pretty cool, in 2020, we started using drones. Another technical name for drones is unmanned aerial vehicles, or you use UAV interchangeably in this presentation. So we use these for mapping habitat, and they're really great. We can send a drone up in the sky, and in this case, you can see in this image here, this drone's just launched from the deck of our boat. So we can go to any habitat in the river and launch this and get a great aerial perspective of our habitat. Prior to this, prior to 2020, we would have to rely on maps, for example, Google Earth and the level of detail that was provided by those. Acquire much higher resolution imagery. And we can see distributions of aquatic vegetation, for example. If we revisit sites, we can also see seasonal changes in the aquatic vegetation. We also have several types of camera payloads on our drone that are sensitive to various wavelengths of light. So we have a camera on our drone that records thermal imagery, so heat signatures, and that's quite useful if you're trying to measure, for example, plumes of different temperature water in the river, other features that might be on the surface that are different temperatures of the water. And we also have another camera that records 5 different bandwidths of light. And that's a lot more than the typical standard camera that would be on your phone, for example. And some of those wavelengths of light and the red wavelength actually allow us to look at plant health because of the reflectance that plants have for those wavelengths. So here's an example. Here's a drone image overhead view of one of our habitats. This is a moat in Akwesasne, and you can see our boat there and some debris in the water. Maybe it's kind of hard to tell, is this a healthy habitat or not? And the next slide is going to show what happens when we look at the different wavelengths of light and how information we can get from those. So here you can see that anything that's healthy green growth is now highlighted in green. So you can see that those submerged logs, for example, are covered in algae, something that we couldn't see in that previous image just with our standard visible light. So this type of camera really allows us to gain insights, and we're able to map out, for example, aquatic vegetation beds that we would only be able to really roughly outline from standing in the river and using a traditional GPS unit, for example. Another super exciting tool that we've added in the past 2 years to the project is a collection of environmental DNA, or eDNA. So you can see some examples here. We have some water samples that are collected, and the way environmental DNA works is that all living things that are in the aquatic ecosystems at some point give off some some genetic material. It could be cells that are just given off by decomposing. Organic matter, for example, also can give off genetic material. All that is just free floating in the water in various concentrations and quantities. And it doesn't last very long in the waters. In some cases, it can only be a few days. And some, if the conditions are right, it can last for weeks. So it really tells you, it's kind of a different way of measuring what's in the water. If we can actually extract that free floating genetic material, amplify it, and look for specific species, for example, or overall genetic material signals for the water. So in our case, collect water samples in the field, and we use controls as well. Controls is a blank water bottle that you bring in the field, and then you would measure back in the lab, and you would make sure that that blank has no genetic material in it. So we did 3 different water collections at each site, just a little spaced apart to give us a little bit of spatial variation in those collections. And in 2021, we actually added the capacity to filter those water samples on site. So it's fresh genetic material. It's filtered filter using a peristaltic pump you can see here. And then we put that in some preservative and freeze that. And then that is then ready to do laboratory analysis at a later time. And I'll talk about that in a little bit. So all this work has allowed us to really focus on species at risk in the river. So these traditional methods we've been using just with our seine net, and now with these new environmental DNA tools, we're able to target various species that we know live in the river. So that's some species that are mentioned here. And another really great aspect is it allows us to track aquatic invasive species, or AIS. And I'll talk about that a little bit as well. So I'll just go through some project highlights now essentially from 2020, incorporating a lot of the overall data set as well. And I just love a shout out to Stephanie Hildebrand, who provided all these amazing and fantastic photos that you're seeing in this presentation, including this one, which is one of my favorites, just show a wetland. And if you remember back to that first map I showed you in 2015, most of our sites are really clustered around Cornwall. And you can see through this time series of maps, and the river has just been oriented vertically now to squeeze all these maps on one slide. But you can see that our project has really expanded in geographic scope, further upstream and further downstream, to the point where we've now expanded into Lake Ontario. This past summer, we were in Prince Edward County, all around the county there. And we've also expanded far down into Lac Saint Louis, and through a great new collaboration as well. So the geographic scope of this project has expanded immensely since it first began in 2015. And all this sampling effort essentially has resulted in one of the largest and most unique aquatic data sets for the Upper St. Lawrence River. We have records now on 157,000 fish species. And I should mention, I kind of always forget the previous slide when I talk about how we catch fish. We put all the fish back. So we capture the fish, we count them, we view them in small viewers where we can look at different morphometric details on them while they're still in water, and then we return them to their habitat. So all these fish do very well after we let them go. Overall, though, we've sampled over 210 sites in the Upper St. Lawrence River, from Prince Edward County all the way to Montreal, essentially. And that includes over 70 wetland species. So we have the health of the wetlands that I mentioned, which are so critical for biodiversity of the river. We've documented over 66 fish species. You can imagine there's quite a bit of skill to identify all those fish. There's over 20 species of minnows alone in the river. Many of those look quite similar. We've collected 65 environmental DNA samples, sampling sites. And that's, again, all the way from Prince Edward County to Montreal. So we have this great data set now in terms of that new tool. And we're also high resolution imagery of all of our sites through our drone work. Our data set includes over 42,000 fish lengths as well. So we have that, we're able to track the progression of size ranges of fish throughout the season. This is a fantastic image by Steph showing a young of the year muskellunge on the top and a young of the year northern pike below. So we can track size development in these species, for example, over the season. One of the great outcomes of this project, and as I mentioned, the River Institute and the Mohawk Council of Akwesasne have been partners from the beginning, is that we've established these amazing data sets for Akwesasne and the surrounding area. So we essentially know at this point what species are present in most habitats that we would find in that area. And because we've been going to many of these sites over the past 7 years, we're able now to start developing analyses of various trends. And as you recall, there's been some really unique events in the last 7 years, including years in 2017 and 2019. So having this baseline data set during these historic flood years has really allowed us to see the effects of those floods on the fish assemblages in the area. Our data, we also come across some really cool things in the river occasionally. This is a bryozoan colony that we typically find in August, during the August survey period, and usually in areas with high nutrients. And this is a colonial vertebrate organism, essentially. It's made up of thousands and thousands of small individual organisms, completely harmless. But I just wanted to show this picture because it's one of the pictures I love, and just to let people know, if you come across one of these bryozoan colonies, while you're having fun in the river, they're completely harmless, and they're native as well. So it's good just to kind of maybe have a look at them, but leave them alone and let them do their thing. One of the things we've learned through all this survey work of the last 7 years is that our nearshore fish populations are dominated by minnows. Are minnows, even though most fish, even sport fish, spend part of their early life cycles in nearshore areas like bass and yellow perch, even walleye. But those species are not minnows. Minnows are a unique family of fish. And as I mentioned, there's over 20 species in the St. Lawrence River, and they generally comprise over 20 to 30% of the overall fish abundance of nearshore areas. And the most abundant minnow of all the minnows is this species you're looking at here called the blunt-nosed minnow. So pretty much anywhere, we're very likely to find this species. This is a male that has some spawning, really cool spawning tubercles on its face. You can see there, those little bumps. We're also documenting the distribution of young of the year northern pike in a lot of our sites. And that's really critical because northern pike populations have declined over the last several decades, likely due to impacts to spawning sites in their nearshore habitats. Part of our work too is because we're measuring, we're documenting fish and also their habitat, plant species, vegetation species that are present, we start to see these fish vegetation associations. And a great example of this is the long-nosed gar that you see kind of hiding amongst these plants here. And this species in particular, we get really excited when we visit a site and we come across lily pads on the surface and tape grass. The combination of those two species generally results more often than not in us finding a long-nosed gar at that site, given the right seasonality of our survey work. That's really neat. You can see the species here perfectly camouflaged in the young of the year stage of its life to blend in with these stalks of these plant species and the leaves of them. So that these plant-fish vegetation associations are really critical because as we develop the strength of these associations, we can start to make predictions about sites that we haven't visited. And that's really key because the St. Lawrence River is enormous. There's no ability of any organization to sample all of it. So it's really pretty key to develop these predictive abilities to say, we haven't gone to this wetland, but based on what we know about sampling other wetlands, we predict we would find the following species there, all things being equal. And then at a later time, we can actually visit that site and test that theory out. If it's a healthy habitat, quite often those predictions are highly accurate. Part of our research too has been heavily focused on the Pugna Shiner. You can see this is a small minnow species. It has a very, very small, see there, almost a pug nose. So it's almost reminds us of Beaker from the Muppets. And we found over 1,000 of them so far in the river at sites that they were previously not known. We've added 20 new sites for the species all up and down the river. So that's been a great result of this project. Young muskellunge populations in the St. Lawrence River really crashed in the early 2000s due to the introduction of an invasive virus, essentially, that affects muskellunge and other species of fish, but in particular muskellunge. So lots of die-off of large adults occurred during that time. And so there's been a rebuilding phase for the population, early 2000s. And part of that has been a really big focus by organizations such as Parks Canada, by Dr. Farrell's group, the Thousand Islands Biological Station, to catalog the locations of young of the year muskellunge nurseries. So measuring where the fish are actually recruiting back into the population. And through our survey work of the northern Canadian portion of the St. Lawrence River, outside of the Thousand Islands, we've been able to document a lot of new sites, 15 new sites where this successfully spawning and the young of the year are surviving. But they remain extremely rare. Only 20 of them have been found in the last, out of 157,000 fish. We get very, very excited when we find one of these fish. You might recognize this fish. Hopefully most people do at this point, if you're an avid fisherman, or if you even just spend a little bit of time in the St. Lawrence River. This is a round goby. It's an invasive species. It comes from the Black Sea, Ponto-Caspian area of Europe and Asia. And it was brought to the Great Lakes in the late 1980s in the ballast water of ships. And it spread following that introduction to all the Great Lakes and the St. Lawrence River. Really, the invasion of the St. Lawrence River was in the early 2000s. And so it's been 20 years, and we now find this fish at almost every single site. Over 90% of our sites, round gobies are present. So it's completely dominated the aquatic ecosystem. In large part, this invasion has happened in tandem with the introduction of zebra and quagga mussels, which are an important prey species for this fish. So essentially, they colonized the river first and created this big food source for this new invasive species to feed on. And these get quite large, and they have impacts for other fish species. They live on the bottom. They're known fish egg predator aggressive for other native species that used to live on the bottom. And they pushed out a lot of those from habitats. So we've been documenting. We also get very excited when we see native benthic species, such as sculpin and log perch, where they would have been occupying habitat that has now been taken over by round gobies. And there is some equalization happening now with the round goby. The numbers aren't increasing as much. A lot of sport fish, for example, smallmouth bass, walleye, have figured out how to feed on these. So they're actually getting quite large based on that. I want to also mention invasive species that we've been tracking. This has been a really important outcome of this project, and based on our geographic range of sampling is this tube-nosed goby. So this is a species that's very closely related to the round goby that invaded in the early 2000s. This species was first detected near the mouth of the St. Lawrence River near Kingston back in 2016. So very new to the river in the last 5 years. And we collaborated with Dr. John Farrell's group in the last few years to document and publish some work on the distribution and expansion of the species in the Thousand Islands. And here you can see that the tube-nosed goby, the upper part compared to the round goby, you can see those tubes kind of sticking out of its nose there. But really easy to identify the species based. You can have a close look at its snout there. Also, the color patterns a little bit different as well. So this is the St. Lawrence River, and you can see these stars represent sites where we located the tube-nosed goby in 2018. So it was very clustered and very limited geographic range in the Thousand Islands. And this is only in 2018. 3 years later, this is where we find all of them now. Just in 3 years, they've been over 100 kilometers in the river. So this is really critical. This is a brand new species that's moving into all these habitats. And prior to 2001, we've had great baseline data sets on what these habitats were like. So we can now compare the changes that might occur once this new species moves in. And again, this is just another layered invasive species on top of the existing brown goby and the zebra and quagga mussels that have also colonized the area. So really significant research, and we're continuing to monitor this species. And it's now, this past summer, the team found some of them in Cornwall. So since 2018, they've moved from the Thousand Islands to Cornwall. As I mentioned, the really exciting new tool that we're using is environmental DNA. And this has been facilitated by a great collaboration with Dr. Steve Lougheed's group at Queen's University. And Dr. Lougheed's research has focused for decades on environmental DNA and biodiversity, and then the use of this tool. So they have a great amount of expertise, and we don't have that background at the River Institute, but we're developing it. So it's been a great collaboration in terms of the River Institute team conducting the field work and then providing collected samples to Dr. Lougheed's lab to conduct analyses. And this has been through some great work by students in the lab and postdocs. So I just want to give a shout out to Stafford Miracle, Oriane Tournier, and Alan Tian for their help on all that eDNA work. All of our 2021 samples have had DNA extracted from them at this point, and there's analyses underway to look at genetic diversity of those samples, as well as targeting specific species within those samples. So screening for species for example, Pugna Shiner or muskellunge. So we're really excited, and results are going to be available very soon from that. Thanks to the work that's going on. We've established through this project, just growing from that first year in 2020 or 2015, when it was just a small team trying to figure things out. We've now collaborated with 6 universities and various other organizations through our research. This has been a great platform to conduct surveys and then compare results and join forces with other groups. And I've just mentioned a few here. They range from Queen's University that I've mentioned, UCAM, SUNY ESF, Clarkson, and a new partnership with McGill University as well. So yeah, we've been able to map out a lot of our muskellunge habitats. I mentioned as well the Clarkson University collaboration. We're publishing a paper right now on mercury contamination loads and brown gobies and yellow perch in wetlands versus non-wetland sites. We have a great new collaboration on the way with the organization at Maitland Tower. So Philip Ling and his group have been fantastic at providing access to the site for our team to visit. And this is near Brockville. And we're developing that relationship and capacity to increase our sampling at that location, which impacted and has a lot of potential for restoration and remediation. This summer, we also started a collaboration with the Environment Protection Office in Ganawage, and we collaborated with them on 2 separate occasions and visited really a fantastic wetland and sampled that. And we're looking forward to expanding this collaboration in the future as well. Some amazing people that work in that office. A lot of the information I mentioned here is feeding into the Great River Rapport, and that's a great previous Science and Nature talked about that. You can find more information about that on River. Essentially, it's looking at ecosystem health indicators of the St. Lawrence River. So you can imagine a lot of the data we're gathering from the FINS project is feeding and informing that rapport. Moving forward, we're taking environmental DNA to the next level. So we're actually developing the capacity to do this environmental research at the River Institute in terms of acquiring equipment and setting up lab space and training people through a great collaboration with Queen's University to conduct this in-house. So really excited about that moving forward. Expanding our use of drones. So we collected gigabytes of data, imagery data, and there's a whole side of that research where you can interpret the imagery and extract additional information out through computer algorithms, for example. And that's things like different habitat features, extensive vegetation, algal blooms, thermal information. And we're also in a collaboration with Dr. Hugh Liu at University of Toronto, who he and his student and his team are developing the ability of drones to fly out over the river and other water bodies and collect water samples and bring them back for environmental DNA collections. So this kind of skips over the necessity of wading into the water using a boat, and really opens up access to so much more access to these water bodies. So that's a great new area of research, and we're really excited to be part of that. So we have a giant data set to work with. We're doing analyses on that. And so watch out for some papers coming out of that. We're doing a lot more outreach moving forward. And we're also preparing for continuing our survey work in 2022 in collaboration with many of the groups I mentioned already. And there we go. Just a big thank you to you can see from our initial group that went out sampling in 2015, our team has expanded greatly. And that's because of all the different elements we've added on to this project in terms of fish and plants and eDNA and team to work with. They're all super enthusiastic. They've all developed great skill sets. They're all young professionals that can now identify fish and plants and fly drones and collect water samples, and just an amazing team to work with. So thank you to everyone that's been helping with that. I know Unique, you're listening in, you've helped out with some survey work as well. Many people at the River Institute have been able to come out, and I'm just listing here over 50 individuals at the River Institute, the Environment Program at the MCA have been able to come out with the project and learn all these new skill sets as well. I just want to mention this too. We developed recently a story map for this project, the FINS project. So you can see a link there. You can find more information. We'll post this on the River Institute's website. And so I encourage people to visit this story map and explore the project, the maps, the photos, more information about the project at your leisure through the story map. Thank you. Just to end with our field crew that I've mentioned, but all the generations of crew going back to 2015. Big shout out to Jenny Lounsbury and Tyler Sunday, who really were at the start of the project and did so much of these collections. Thank you to the supporting staff at the River Institute. There's a lot of administrative support. Pam Maloney at the Institute has helped me write all of the funding applications that have helped support this work. So thank you, Pam, and others. All of our collaborators, all of our funders, we have amazing funding support from different sources. Thank you, Stephanie Hildebrand, for your amazing photos. Thank you, Emily Cormier, for helping put together this presentation as well. And I just wanted to end on this very last slide. This is one of my favorite pictures actually from this past summer. And we just completed a survey, and it was kind of a hot day, and the crew just kind of sat down in the river and they just started to look at stuff. And this is the kind of person, the kind of people you want on your crew, the people that even after work is done, they just want to sit and look at things. And I think at the time we were looking at fossils, but other small fish and invertebrates and whatever else we could find. There's such a keen interest in learning about the river, and I just love what this picture conveys. You know, that initial interest that the River Institute and the Mohawk Council of Akwesasne had at the beginning of the project has just grown in scope and incorporated so many new people, and we really look forward to continuing that in the future. So thank you very much.
Excellent. Thank you so much, Matt. That was a fantastic presentation. What a great slide to end on. Also, just that's such a great photo to see everyone out in the field like that. Curious minds, you know, really inquiring even after the field work is done. So we'll move on to questions now. So this is the question and answer period. We would invite anyone who has any questions for Matt. If you would like to post them in the comment section on Facebook. I see that we already have a few questions. And we have also shared the story map for the FINS survey. So that's also posted in the comment section if anybody would like to see. So for a first question, Matt.
Thank you for sharing that.
Yeah. Someone has asked, are there many fish species that stay in the deeper water that are not sampled along the shorelines?
That's a great question. Because for sure, we're not capturing the complete diversity of the river by just focusing on the nearshore areas. There's some species, for example, of sculpin that inhabit the deeper waters and rarely come into shallow waters. Other species we're just not going to find in our survey. American eels, extremely endangered in the river, but we wouldn't training techniques, sturgeon as well. Other cold water fish that prefer cold water or cooler waters are going to be inhabiting the deeper waters of the river. And interestingly enough, I mean, there's a great smallmouth bass population in the St. Lawrence River that's, you know, a lot of sport fishermen are commenting on the larger sizes that are being captured in the last couple of years because they're feeding on gobies, but we actually rarely come across young of the year smallmouth bass. It's much more common to come across the largemouth young of the year, just because the smallmouth are a little more distributed in deeper waters at those younger life stages. So certainly that's a great question. This type of survey work needs to be combined with other techniques, and there are other surveys conducted by government agencies that use gill nets, for example, that target larger fish in the river. So that type of survey data combined with our nearshore techniques and the nearshore techniques of other organizations really give you that complete picture. And the really exciting thing is the new use of this environmental DNA tool, which has the potential to cover everything. It really depends on the water currents and particularly in a river, but it kind of skips over that gear limitation. Whatever's floating in the water, we should be able to identify.
Great, thanks, Matt. So we have another question here. It says, I am used to thinking of nutrient runoff as being bad for aquatic habitats, but are there fish or other marine creatures that thrive around areas of high nutrient runoff?
That's a great question too. We definitely see, in some cases, the nutrients that flow into the river encourage more plant growth. And more plant growth generally creates more food sources, more cover for fish, especially small fish and young of the year fish. So in those types of environments, and they tend to occur near the mouths of small tributaries entering the St. Lawrence River. A great example is in Bay St. Anisette, the back side of Lake St. Francis. There's a small tributary that Rivière de Guerre that comes out there, I believe that's the name of it. And lots of nutrients are flowing out of that. And that's one of our highest biodiversity sites of any of our sites that we surveyed. In some occasions, we catch over 20 species of fish in one visit. So certainly the nutrients can create conditions that would encourage biodiversity. And that's not necessarily a bad thing. It's when the nutrients are in excess. So too much nutrients is always a growth. And we do, again, in many of our sites in the river, again, typically near the mouths of tributaries, but other areas as well, we'll see the large algal mats just floating on the water. Our nets get fouled by it. The algae is not just on the surface, but also on the bottom of the river. And different types of algae create different difficulties for our crew. But it's certainly later in August, we tend to see that. And in some cases that those algal growths impede fish health. So they essentially take over the habitat. It's sometimes difficult to see from the surface, but in a lot of cases that's covering the bottom, and fish can get stuck in that, small fish can get stuck in it, and it also can severely decrease the oxygen in the water. So I would say nutrients are perhaps necessarily bad earlier in the season, but later in the season, when in combination with warmer temperatures of the water, it can create really bad scenarios for algal blooms.
Okay, perfect. Thank you. I don't see any more questions so far in the Facebook comments, but I do have a question for you. I'm curious, do you happen to know what the lifespan is of sorts for eDNA? And if perhaps these samples would be able to identify environmental DNA of species that used to exist in the St. Lawrence River and that are now extinct?
Yeah, that's a good comment. So free-floating eDNA, it depends on a lot of conditions. If the water is really warm, the DNA is going to decompose, deteriorate more quickly. If there's UV light, for example, that can also degrade it. And also the amount of oxygen in the water is known to affect that as well. So it's kind of hard to predict how long it's going to last. Sometimes it can be on the order of several days. If conditions are right, it can actually last for weeks as well. There is some projects that are being developed at Queen's University by some students there that are going to be, that's Stafford Miracle, where they're going to be looking at cores of sediment and looking at the environmental DNA, going back in time in that core, see what types of information they can find. So stay tuned for that. That's a very cutting edge research project on the go.
Excellent, thanks, Matt. We have another question here. Any sign that water temperatures are increasing?
I would say on average, I mean, we've only been doing our survey for 7 years. If you look at the data going back decades and decades back, there is a slight trend in warming, and we also see that too, right? Our seasons are getting our warm summer, spring, fall seasons are getting longer. Our winter seasons are getting shorter. In general, there's less ice. I mean, you have to look at long-term patterns, but even just in the last few years, we've had less ice than previous years. So all that does affect the populations, and ice cover is critical for some aspects of life cycles of fish. It creates protection and capping on them. It also affects the growth of vegetation, for example. So the critical thing, and it doesn't really happen in the St. Lawrence River because the water is really well mixed, but in conditions where you have lots of warmth, water will stratify, thermal stratification. And when that sets up, the water column doesn't mix very much. And so what happens on the very bottom layers that aren't mixing with the surface where oxygen is in the atmosphere is that they can become depleted of oxygen. And so you can get these scenarios where you have very low oxygen, anoxic conditions, which can be very harmful for fish. So yeah, the warming trend in general, it will favor some fish that are suited to live in warmer water. It might affect some invasive species that maybe aren't a problem now, but will become a problem in the future. And there are some researchers looking at that. Our collaboration with Dr. Tony Ricciardi at McGill University, we're collaborating and looking at how Goby and Round Goby will respond to climate warming scenarios. And so they're conducting some laboratory tank analyses under various conditions to look at that question.
Excellent, excited to see what those results are.
Mm-hmm.
So we have a few more questions, Matt. The next one is, do the FINS nets ever find things like salamanders, mud puppies, and other amphibians in your studies?
I don't think we've ever caught a salamander with our net, but we do occasionally cross them in the habitats. And a big shout out to Kate Schwartz on our team that is the world expert as far as I'm concerned in finding salamanders and amphibians and reptiles. So if they're around the habitat, Kate will find them. So we do come across them, and the most common one is the mud puppy. So it's the largest salamander species in Ontario and fully aquatic species. So it makes sense that that would be the one that we come across when we're doing aquatic surveys, and they can get quite large.
Perfect. We have another question. It says, based on your surveys over the years, how healthy is the St. Lawrence River, and what is better and what is worse?
I think I missed the last part of your question there, Unique, sorry.
Yeah, sorry. So based on your surveys over the years, how healthy is the St. Lawrence River? What is better and what is worse? I'm assuming this is maybe to like ask which parts specifically of the St. Lawrence River are healthier or less healthy. Oh, Matt seems to have frozen. All right, hopefully come back soon. Okay, Matt is still frozen, so we'll just wait a little bit until he comes back. Oh, there you are. Perfect.
Sorry about that. I got dropped there.
No
I'll say
worries.
really quickly, this is a great question to flip to the River Report team, who've been looking specifically at that question. What is the health of the St. Lawrence River? And it really depends on your perspective. We're focusing on fish, but there's lots of other aspects of the river. Water temperature, vegetation, plants, water quality. They have dozens of indicators they're looking at actually. So I would encourage you, there's no simple answer to that question. I would say that, just documenting what we've seen through this project, the river has an incredible amount of diversity and some amazingly pristine habitat still. There's not a lot of them left, but there's still some there. Akwesasne being a great example that some of the giant wetland complexes there that still have amazing diversity. Great places in the Thousand Islands as well. So we are documenting positive things through this survey. Trend-wise, it's I don't think we have the long-term perspective to be able to answer that question. But again, just to encourage you to look into the River Rapport, the Great River Rapport outcomes and outreach to maybe find some more information on that question.
Great, thanks, Matt. I'm also just going to take a really quick second before we continue with the questions to thank Gabe from Your TV, because we've hit the 1 hour mark. So if Gabe, you need to log off, just thank you to everyone who watched this through Your TV. We will continue on with the questions through Facebook Live. So we have another one here, which says, any comment to make on the attempt to introduce salmon breeding in Lamprey Park?
Yeah, I think, so going back to the 1950s, 1960s, we had introduction of Pacific salmon to the Great Lakes. And, there's a lot of avid fishermen that love fishing for Chinook salmon and coho salmon and other rainbow trout, for example, to introduce. But these are not native fish species to the Great Lakes systems in the St. Lawrence River. The native Salamonid species is the Atlantic salmon. It's been extirpated completely from the St. Lawrence River. So if there is a focus on reintroductions, it really should be on reintroducing native species like the Atlantic salmon. And that would be the only one I would support from a scientific perspective and ecological perspective.
Okay. One more question here. So someone has asked, have you ever found any home aquarium fish like pleco or goldfish in your catches? And what is the most surprising species found?
It's an interesting question. We haven't. Luckily, we haven't found any aquarium fish at all. If you were to go to the harbor, you are very likely to come across goldfish. So there's a thriving goldfish population that is present there now that is 100% due to releases from aquaria. But we don't find that in the St. Lawrence River, luckily. And goldfish in general, I mean, they're not ideally suited to survive in cold temperatures, but in that case, they've found a way to reproduce. But yeah, no plecos, no other species like that, thankfully. We have another species that's invasive that's moving in upstream from Montreal right now is the tench, which is a minnow species. We are finding them in Lac Saint Louis, but we haven't come across any yet in Lake St. Francis. So we'll be on the watch for those as well. And again, that was an introduced species going back to the 1980s. And a lot of government agencies in Quebec are tracking their expansion there. The most interesting fish we've caught, I mean, one of my favorites is, I have a couple favorites, young of the year muskellunge. That is the most excited fish to catch in the survey because they're so rare. Only 20 of them out of 157,000. We love catching bowfin, which is another fairly rare species. And then long-nosed gar are always kind of fun to catch as well. And just really cool adaptations that all those fish I just mentioned have to survive in these nearshore areas.
Excellent. I think this goes well into the next question. Someone has asked, are there many lampreys?
Yeah, we don't. So lampreys spend most of their life cycle in the juvenile stage, and they kind of bury themselves in sediments and filter feed, and then at the very last stages of their life, they eventually do an stage. Some forms of those adult stages are parasitic and will attach to fish. Others are just reproduce and die. So the type of survey we do with the seine net, we're very unlikely to come across free-swimming lampreys or those larval stages of them. We do occasionally come across them when they're attached to a fish. So we've come across them attached to a northern pike and carp. And in those cases, the species have been identified as silver lampreys. Now, interesting thing is silver lamprey is actually a species at risk. So it's not a bad thing. There's actually not that many of them that are known. I know there is some concern occasionally. You may see wounds on fish, for example, smallmouth bass, that correspond kind of the parasitic attachment location of a lamprey. We unfortunately we can't track that group of fish through our survey work. It's really interesting to think about their role in the ecosystem. And the invasive sea lamprey in the Great Lakes in particular has caused a lot of damage to lake trout populations. And there's huge efforts in the Great Lakes to control sea lamprey populations through essentially a very targeted into streams where they spawn and where those larval stages develop. You can specifically target them with chemicals and kill them off essentially. We don't, as far as anyone knows, we don't have a sea lamprey invasion issue in the St. Lawrence River though.
Great. We have another question, Matt. Someone has asked, any sign of invasive species from the Mississippi watershed?
I mean, invasive species covers a pretty broad range of taxa, and I've just been focusing on fish. So in terms of fish, we haven't seen. We have, I mentioned a few that the invasives we are tracking. We do come across quite a bit of invasive plant species. It's quite widespread in the river now. European milfoil, curly-leaf pondweed, to name a few, various types of invasive algae. So those types of taxa and species we are documenting in our survey work. There are other invertebrate species, so rusty crayfish and others that are known to be expanding in Ontario. We haven't come across any in our survey. We do take note of, say, crayfish, for example, that we might pick up in our seines, which was pretty common actually. But in terms of other invertebrates, we're not really. We haven't really noticed an invasion of any new species.
Okay, excellent. I don't see any more Facebook questions for the time being. If it's okay, I would like to ask one more, maybe just to finish off for the night, if that's okay.
For sure.
I'm just curious, if people have waterfront properties, what might they be able to do on their own properties to benefit these fish populations?
Yeah, there's a lot of, great question. There's a lot of efforts by various organizations, Watersheds Canada, various conservation authorities to try to encourage waterfront owners to essentially naturalize or even just soften their waterfronts. And I understand the tendency of a lot of people is to clear vegetation along your waterfront so you can enjoy the view of the river. That's an understandable thing to want to do. But there's different ways of going about that where some ways of clearing all the vegetation and having grass, for example, right up to the shoreline. You're essentially removing all of the anchoring roots and vegetation that maintain that shoreline. Now trade off for that view from your property. And so that destabilizes the nearshore area, creates erosion, sedimentation. Quite often you see a disappearance of aquatic vegetation offshore as well as part of that. And then you see a decrease in plants and other interesting things to look at. And there's been some great, I was on a, Barbara King from Watersheds Canada gave a great presentation recently where she was talking about some examples of Some of the work they've done with some property owners to renaturalize their properties, plants native species of vegetation back. And over the course of several years, they've just been able to witness incredible growth and biodiversity return to these properties. And in some cases, they've become the focal point of the neighborhood for the kids to come visit, because that's where all the cool stuff is. It's not along the barren, grassy waterfront for a lot of these properties. It's where all the plants and the complexity of the environment is. So the kids, follow the kids. They know what's right, I think, and hopefully they learn from that too, and maybe can encourage their parents or their neighbors or other people, as well as all these other organizations to renaturalize those habitats. You don't have to do the entire thing. You could do little pieces of your habitat to start with, your shoreline, to see how that works. But just to let you know, there is subsidies and funding available to help people do that if you're interested. So you don't have to bear the entire cost to do that. There's a lot of support for that.
Perfect. Thanks, Matt. I do think that's all of the questions for tonight. So thank you so much for that excellent presentation. Big thank you also to Zach for making sure that everything goes smoothly. Yeah, Matt, this was very informative. We're very thankful to have you at the River Institute, and you and the FINS crew and all the great work that you do. So thank you. Yeah, so that'll be it for tonight. We would like to invite everyone to join us next month on January 5th for the next Science and Nature Untapped. And all of these videos will be recorded, saved to our Facebook page, as well as River Institute Untapped. So thanks for joining us, everyone.
Thanks, Nick.